Hello students, welcome to today's lesson. By the end of today's lesson, you should be able to differentiate between test cross and then back cross. Then you should also know about monohybrid inheritance in humans, then non-Mendelian inheritance in humans. Come along. Now, let's talk about back cross. When we talk about back cross, you can say that a back cross is a cross between the offspring, that is usually the first filial generation, and one of its parents, or an organism that is genetically similar to the parent. Now, why do we carry out a back cross, or why do we do a back cross? Back cross is carried out to retain or study the parental traits. This is a genetic cross representing a back cross. So these are the parents and then the first filial generation. So using this first filial generation to cross the parent, that's the first filial cross with what? The homozygote parent, that's the back cross. Let's talk about a test cross. When we talk about a test cross, we can say that it's a cross between individual showing a dominant phenotype and another individual that is homozygote recessive for that trait. Why do we carry out a test cross? We carry out a test cross to determine whether the individual is homozygote dominant or heterozygote. So this is example of a test cross. So we have the dominant or the homozygote dominant, which is tall. We are crossing it with a homozygote recessive, which is a dwarf. So after the cross, you realize that all the offspring or all the four offspring are tall. There is no short. So the second cross, that's the heterozygote, is also physically tall. We are crossing that one too with what? Homozygote recessive. Then after the cross, all the offspring, you realize that two of the offspring are tall, and then two of the offspring are also short. So this is how a test cross is done. Let's talk about monohybrid inheritance in humans. In our previous lesson, we talked about monohybrid inheritance, where we said the transmission of one trait from parent to their progeny. So when we come to the monohybrid inheritance in human, now, if the trait is dominant, that means both individual with homozygote dominant and then the heterozygote gene will show the trait. That is, we have example to be tongue rolling. Then if the trait is recessive, only individual with homozygote recessive gene will be able to show the trait. Example is what? Albinism and then color blindness. Let's use this diagram to illustrate the monohybrid inheritance in humans. So we have, when it comes to tongue rolling, we have these two parents. Though they are all heterozygotes, they can roll their tongue. So the probability that their offspring, or the probability of their offspring are, we have one homozygote dominant, two will be heterozygote, and then one will be homozygote recessive. Now, both the homozygote dominant and then the heterozygote can roll their tongue, whilst the homozygote recessive cannot roll its tongue. Why? Because the trait for tongue rolling is a dominant gene. Now, let's come to that of albinism. Now, when we talk about, let's say, two parents who are carriers of albinism, the probability that the offspring or the probability of their offspring are: one will be homozygote dominant, two of them will be heterozygote, then one will be recessive. The recessive one is going to show the trait for albinism. Why? Because the trait for albinism is found on the recessive gene. Now let's look at non-Mendelian inheritance. When we talk about non-Mendelian inheritance, you can say that they are inheritance that do not follow the pattern of Mendel's experiments. Now, Mendel's proposed of two alleles, which are the dominant allele and then the recessive allele. The dominant always marks the effect of the recessive allele. But when it comes to this case, it's not like that. That is, or this non-Mendelian does not follow the pattern of what Mendel proposed. So we have example of the non-Mendelian inheritance, or some of the examples are: we have incomplete dominant, we have co-dominant, we have multiple alleles, then polygenic inheritance or polygenic trait. Let's take the first example of non-Mendelian inheritance, which is incomplete dominance. When we talk about incomplete dominance, we can say that it's a situation whereby an allele expresses itself partially in heterozygote state. So, for example, let's take these flowers. We have a red flower and then a white flower. The outcome is supposed to be either a red flower or a white flower, but in this case, that is in Mendel's inheritance. But when it comes to this case, you realize that we have a pink flower here, which does not follow Mendel's experiment. So an example of incomplete dominance in human is an individual with the sickle cell trait. So let's take these two parents. One is a sickle cell, or one is carrying the trait for sickle cell, that is AS, and then one is what? A normal person. That means this person or this parent is not suffering from a sickle cell. Now, the probability that their offspring, or the probability of their offspring, are two of the offspring are going to be carriers of the sickle cell, that is the AS and then the AS. Then two of the offspring are also going to be normal children. Now, when we take the AS, you realize that none of the allele fully or totally dominate over the other. The A is showing and then the S is also showing in the individual. So, individual with the AS will suffer from mild symptoms of the sickle cell. Now, let's talk about co-dominance. When we talk about co-dominance, we can say that it's a situation where two alleles express themselves equally in a cell or organism. So let's take example, or an example to be the blood type AB. With the blood type AB, we have the A having for the antigen A, and then we have the B to also be having the antigen B. So both the antigen A and then the antigen B, they express themselves equally in the blood type AB. Another non-Mendelian inheritance is the multiple allele. When we talk about multiple allele, it's a situation in which a trait is controlled by more than two alleles. Example is the ABO blood group system. Now, the ABO blood group system is controlled by three different alleles. Now, we have allele allele for blood type A, we have allele for blood type B, and then we also have allele for blood type O, which are the 3 alleles controlling one trait. Now, when we take blood type A, someone with blood type A is either having 2 alleles, that is AA alleles, or is having 1 A allele with 1 O allele. So we can refer to them as someone who is having blood type A. Then someone with blood type AB is either having or is having the A allele and then the B allele. Both the allele are present. Then when we talk about someone with blood group B, this blood type B is either having the B allele or 2 B alleles, or is having 1 B allele with O allele. Then someone with blood type O is having O allele or 2 O alleles. So this is the multiple alleles. Polygenic inheritance is another non-Mendelian inheritance. When we talk about polygenic inheritance or polygenic traits, we can say that they are those traits that are controlled by 2 or more genes. For example, we have weight, height, skin color, eye color in humans. So when you look at this diagram, you realize that we have a lot of genes controlling, or more than 2 genes controlling one trait. That is the trait for eye color or the trait for height. So that's the polygenic inheritance. Now, before we meet in class, try your hands on these questions. What is the significance of a test cross? Define albinism. Then you solve this. A man who is heterozygote of the allele for albinism married a woman who is also a heterozygote of the gene. What is the probability of their offspring having albinism? Then differentiate between incomplete dominant and co-dominant.